Oh, Bismillahirrahmanirrahim. Hello, everyone. Our today's topic is phospholipids or complex lipids and ARDS, that is acute respiratory distress syndrome. Let's first start with the phospholipids. What are phospholipids or what are lipids? Lipids are organic greasy compounds which are insoluble in water and soluble in organic solvents like ether, chloroform, benzene, carbon tetrachloride, etc., etc. So lipids, on the basis of their chemical structure and composition, they are divided into various classes. Uh, Simple lipids, compound lipids, and derived lipids. Simple lipids are they are the esters of fatty acid with alcohol, glycerol, or waxes. Again, the neutral lipids or the simple lipids are of two types. One are the fats and the other ones are the oils. Beeswax is a ester of fatty acid with a long chain fatty acid. Uh, long chain fatty acid. Then compound lipids, these are the lipids which contain, in addition to alcohol and fatty acid, some other additional molecules such as phospholipid phosphate or phosphoric acid, and along with the uh, glycerol and fatty acid. Then we have the glycolipids, in which the carbohydrates are present in the lipids, and the phospholipids are of two types, depending upon the type of the alcohol that they contain. That is the glycerophospholipid, those phospholipids which contain the glycerol as alcohol. Then sphingolipids, these are the ones which contain the alcohol, sphingosine. This tells you even slightly better picture. The complex lipids, phospholipids, and glycolipids. As the name suggests, phospholipids are the ones which contain the phosphoric acid and the phosphate in their structure, and glycolipids are the ones which contain sugar in their structure. Now, phospholipids are divided into two groups on the basis of the type of alcohol that they contain. That is glycerophospholipid having glycerol as the alcohol, and a sphingophospholipid containing sphingosine as an alcohol, which is a 18-carbon unsaturated amino dihydroxy alcohol. Glycolipids are the ones which are also sphingolipids containing different type of sugars. Now let's discuss with simple lipids. Simple lipids are, as you can see right here, this is the glycerol attached with 3 fatty acids in the ester linkage, and these are known as triacylglycerol or triglycerides. Coming to the complex lipids, one type are glycerophospholipids, ether glycerolipids, and sphingolipids. You can see right here, the glycerol is attached to 2 fatty acids, and other than 2 fatty acids, we have a phosphoric acid right here, and then has a head group. So that makes the glycerophospholipid. And we have different type of head groups, therefore we have different type of phospholipids, glycerophospholipids. Ether glycerolipids are those in which at carbon number 1, instead of having a fatty acid in the ester bond, we have a unsaturated fatty aldehyde in the ether linkage. The rest of the structure is same. And then sphingolipids are the ones in which we have the sphingosine as the alcohol, which I mentioned is the 18-carbon amino unsaturated dihydroxy alcohol. And this contains the alcohol, fatty acid, and the phosphoric acid, and a head group. Coming to glycophospholipid, this is the general structure of the glycophospholipid. You can see right here, this is the polar group, which is the nitrogenous base. This is the phosphate, another part of the polar group. And then the rest of this is the hydrophobic tail of the fatty acids, which is a hydrophobic in nature. So it has a hydrophilic head and hydrophobic tail. That's why phospholipids are known as amphiphilic in nature. Amphiphilic in nature. As far as the biosynthesis of glycophospholipid is concerned, the first step of glycophospholipid synthesis is common with the pathway to triacylglycerol, in which two fatty acid groups are esterified to carbon number 1 and carbon number 2 of the L-glycerol 3-phosphate to form the phosphatidic acid. That's what is phosphatidic acid. Phosphatidic acid is the one which contains glycerol attached to two fatty acids and a phosphoric acid. This phosphatidic acid is a parent compound of all the glycerophospholipids. Again, remember that this phosphatidic acid is the parent compound or the compound of all the glycerophospholipids. That is, all glycerophospholipids contain in their structure the phosphatidic acid and plus different type of nitrogenous polar compound. Now, glycerophospholipid, depending upon the type of nitrogenous base attached to phosphatidic acid, we have a different type of glycerophospholipids. For example, if serine is attached to phosphatidic acid, it is known as phosphatidylserine. If ethanolamine is attached to phosphatidic acid, it is called phosphatidylethanolamine. The common name is cephalin. If choline is attached to phosphatidic acid, it's the phosphatidylcholine. The common name is lecithin. And this phosphatidylcholine is very, very important because we will be discussing later on in case of the respiratory distress syndrome. Then inositol attached to phosphatidic acid results into phosphatidylinositol. And if glycerol is attached to phosphatidic acid, it is called phosphatidylglycerol. This is the common structure of the phospholipids. You can see right here, this portion, this one fatty acid, another fatty acid attached to glycerol, and a phosphate is the phosphatidic acid. And you can see right here, we have a serine as a nitrogenous compound. Therefore, it is known as phosphatidylserine. This one, instead of serine, it has the ethanolamine. Therefore, it is known as phosphatidylethanolamine. But the common name is cephalin. Phosphatidylcholine, you can see right here. This one, the two fatty acids attached to the phosphoric acid, the phosphatidic acid, and the head group of the nitrogenous base is the choline, which is a trimethyl nitro compound. And its common name is phosphatidylcholine. Cardiolipin, another glycerophospholipid in which the two phosphatidic acid are present, and in the center of the two, we have a glycerol. You can see right here, the one phosphatidic acid attached to one carbon of the glycerol, and the second phosphatidic acid attached to the third carbon of the glycerol. So this is known as the cardiolipin. And this cardiolipin is distributed in the inner mitochondrial membrane. Function is the maintenance of respiratory complexes of electron transport chain. Ether glycerolipids. As I pointed out, ether glycerolipid contains at carbon number 1, instead of having the fatty acid and ester bond, it contains the unsaturated fatty aldehyde in the ether bond. The rest of the structure is same as of the glycerophospholipids. The common example is plasmalogen. You can see right here, this is the ether linkage right here, unsaturated fatty aldehyde, and this is the acyl group, fatty acid, phosphoric acid, and the nitrogenous base ethanol amine. So this common name is plasmalogen. Phosphatidylethanol present in nerve tissues and phosphatidylcholine in heart muscles. Their function is more resistant to oxidative stress, therefore provides protection to tissues with active aerobic metabolism. Platelet activating factor. There is another ether containing phospholipid in which at carbon number 2, instead of having a long chain fatty acid, we have only acetyl group, acetic acid. And the rest of the structure is same. That is the phosphoric acid right here and the nitrogenous base, ethanolamine. Their function is released by a variety of cell types, including platelets, neutrophils, basophils, and endothelial cells. The function is it activates inflammatory cells and mediates hypersensitivity, acute inflammatory, and anaphylactic reactions. Phosphatidylinositol is another important phospholipid which is mostly present. About 10% of the total phospholipid is present. Phosphatidylinositol is present in brain. Its structure is attached to phosphatidic acid. We have a hexahydroxy cyclic alcohol. You can see right here, the 6 carbon, 1, 2, 3, 4, 5, 6 carbon, all of them contain the hydroxyl group, and which is esterified to the phosphoric acid. It's carbon number 1, it has stearic acid, 18 carbon, hydrocarbon, saturated fatty acid, and carbon number 2, it has arachidonic acid, which is unsaturated essential fatty acid. This is one of the essential fatty acid. The function of phosphatidylinositol is cell signaling, reservoir of arachidonic acid, and protein anchoring. Remember that the arachidonic acid is a precursor for the synthesis of the active hormone prostaglandin. Phosphatidylinositol 4,5-bisphosphate. Now let me remind you that 4,5-bis, although it has 2 phosphate at 2 different carbons. Now if the phosphates or any additional group is present more than 1 at the same carbon, then it is known as bi or di, and if it is attached to the 2 different carbons, then it is known as bis. So in this case, you can see right here, the phosphate is attached to the carbon number 1, and another one is carbon number 2. There are 2 different carbons, and therefore, sorry, carbon number 4 and carbon number 5. And therefore, we can call it as a bisphosphate. So, phosphatidylinositol 4,5-bisphosphate. It is an unusual phospholipid in that it often contains stearic acid at carbon number 1 and arachidonic acid at carbon number 2 of the glycerol. It therefore serves as a reservoir of arachidonic acid in membrane and provides substrate for the synthesis of prostaglandin. Sphingolipids. This is the second type of phospholipid in which instead of having a glycerol as an alcohol, it contains sphingosine as an alcohol. Sphingosine, as I pointed out, is 18 carbon unsaturated dihydroxy amino alcohol. To sphingosine, fatty acid is attached, the phosphoric acid is attached, and a head group is present in this one. Look, this is the structure of sphingosine. You can see right here, 1 alcohol, the second OH group, amino group, and the unsaturated double bond right here. So it is 2 amino, 4 octadecene, 1,3-diol. sphingomyelin. Sphingosine, which is this is the structure of the sphingosine, okay? And then right here is an nitrogen base, and there is a phosphoric acid. Now, sphingosine plus fatty acid. Sphingosine plus fatty acid like this, you can see right here. One fatty acid, two fatty acid attached to the sphingosine. Then this is known as the ceramide. So ceramide is a parent compound of all the sphingospholipids. Different type of sphingospholipids differ from each other on the basis of the type of nitrogenous base which it contains in its structure. You can see right here, ceramide attached to phosphoric acid, then attached to choline as a nitrogenous base. And the common name of this compound is a sphingomyelin. It is distributed and the constituent of the myelin sheath of nerve fibers. You can see the name from the name sphingomyelin present in the myelin sheath of the nerve fibers. They are building block of the myelin sheath, primary source of ceramide and signal transduction. Recall again, the ceramide is the one in which fatty acids are attached to sphingosine, just like phosphatidic acid, in which the fatty acid and the phosphoric acid attached to glycerol, which is the parent compound of all the glycophospholipids, and ceramide is the parent compound of all the sphingospholipids. Degradation of phospholipids. Phospholipids are degraded by 4 different enzymes, which are specific for a specific carbon number. The one is the phospholipase A that removes the fatty acid at carbon number 1 of the glycerol, and the phospholipase A2, one is the phospholipase A1, and the second one, phospholipase A2, which removes the fatty acid attached to the carbon number 2 of the glycerol. And the third is the right here. We have 2 different. One is the phospholipase C, which hydrolyzes the phospho fatty acid, phosphorylated compound. And if it is attached from attack right here, it's only removal of the nitrogenous base, then it is done by phospholipase D. But this one, phospholipase A2, is very, very important because it results in the formation of the lysolecithin, which is very poisonous and which is present in the snake venom, and it has a hemolytic activity. So that's why when a snake bites, then it results into lysolecithin, and which results in the hemolysis of the red blood cells. Niemann-Pick disease. Niemann-Pick disease is due to the deficiency of the enzyme sphingomyelinase. Let's see what sphingomyelinase does. This is the structure of sphingomyelin. You see right here, this is a ceramide, fatty acid, and the sphingomyelin. Right here, phosphoric acid, and the choline is attached to this one. So if right here, 2 different enzymes degrade the sphingomyelin. One is the ceramide, ceramidase right here, means because it is degrading the ceramide, and recall that the ceramide is sphingosine plus fatty acid. So ceramidase removes the fatty acid from the sphingosine, and the sphingomyelinase removes the phosphorylcholine right here. You can see right here, this one, the phosphoric acid, phosphate, and a choline, phosphorylcholine. So because of this deficiency of this enzyme, then what happens? The sphingomyelin keeps on accumulating and results into enlarged liver and spleen, which is filled with lipids. Results into severe mental retardation and neurodegeneration, and then death occurs in early childhood. It is the autosomal recessive disease, inability to degrade sphingomyelin, deficiency of sphingomyelin A, which is a type of phospholipase C. Glycolipids or glycosphingolipids. Look at this one, sphingosine plus fatty acid is a ceramide. So to ceramide, if carbohydrates are attached, then they result into sphingoglycolipids. Or in other words, they are derivatives of ceramide containing long chain fatty acid and a sugar molecule. Different type of glycolipids differ from each other on the basis of the type and the number of sugar molecules attached to the ceramide. We have 4 types of different glycolipids: cerebrosides, globosides, gangliosides, and sulfatides. Their distribution is essential components of all membrane in the body, greatest amount in nerve tissues. Function is regulation of cellular interaction, growth and development, body blood group antigens. Now coming to cerebrosides, ceramide monosaccharide. Cerebrosides are ceramide monosaccharide. That is, monosaccharide sugar is attached to ceramide. It is the simplest neutral glycosphingolipids. And if galactose is attached to ceramide, then it is known as galactocerebroside, the most common cerebroside found in the membranes. Another one, if glucose is attached to the ceramide, it is glucocerebroside, serve as primarily as an intermediate in the synthesis and degradation of more complex glycosphingolipids, which contain more than 2 or oligosaccharides. Cerebroside are found predominantly in brain, but 70% of dry weight of brain is cerebrosides, and peripheral nerve tissues with high concentration in the myelin sheath. Galactocerebroside, this is the structure you can see right here. This is a ceramide, and the galactose is attached to this one in glycosidic linkage. Globosides are ceramide oligosaccharides. Addition of monosaccharides, including galactose and acetyl galactosamine to glucocerebroside. You can see right here, cerebroside, glucose, galactose, it is known as lactosyl cerebroside, lactosyl ceramide, cerebroside, glucose, galactose, and acetyl galactosamine, which is commonly known as the Forceman antigen. Gangliosides, they are negatively charged at physiological pH, but look right here in the structure, there is a ceramide containing several sugars: glucose, N-acetyl galactosamine, galactose, and N-acetyl neuraminic acid, which is the common name of sialic acid. So glycolipid containing sialic acid and acetyl neuraminic acid, found primarily in gray matter of brain, ganglion cells, and central nervous system, particularly at the nerve endings. Also act as cell membrane receptor. Gangliosides, the nomenclature, is based on the number of sialic acid residues present in the molecule. It may contain 1, 2, 3, or 4. On the basis of this, we may have the GM1, GM2, GM3, and so on. So GD means 2, GT3, and G4, GQ4, 4 sialic acid residues in the molecule respectively. On the sequence of the carbohydrates, The number after the GM, that is GM1, for example, refer to the structure of the oligosaccharides. These numbers were derived from the relative mobility of glycolipids on thin layer chromatograms. The larger GM1 gangliosides migrate the most slowly, then the next larger, and the next one, and the next one, and the smallest one will move faster. Gangliosidosis, it is an inherited lysosomal disorder which progressively destroys the nerve cells in the brain and spinal cord. Sulfatides, cerebroside that contains sulfated galactosyl residue, negatively charged at physiological pH, found predominantly in nerve tissues and kidneys. Sphingolipidosis, defect in sequential degradation of glycolipids, lead to a number of lysosomal storage diseases. Sphingolipidosis or cerebrosidosis or gangliosidosis. A specific lysosomal hydrolytic enzyme deficiency is present in each order. Therefore, usually only a single sphingolipid, the substrate for the deficient enzyme, accumulate in the involved organ in each disease. Gangliosidosis, GM1, inherited lysosomal disorder, which progressively destroys nerve cells in the brain and spinal cord. This is the chart that shows you a different type of diseases or different type of, because of the gangliosides. Look at right here, how many sugars that we have? 1, 2, 3, 4, 5. If the terminal sugar, which is galactose, it is removed by the beta-galactosidase. If this enzyme is deficient, then the GM1 is normally accumulated, and the disease is GM1 gangliosidosis. The next one, and if this enzyme is not deficient, then we get the N-acetyl galactosamine, galactose, the glucose, and N-acetyl neuraminic acid right here, attached to ceramide. So the N-acetyl galactosamine is hydrolyzed by beta-hexosaminidase enzyme or hexosaminidase A. And if this enzyme is deficient, then the ceramide containing 4 sugars up right here cannot be broken down, and we get the GM2 gangliosidosis, and the common name is Tay-Sachs disease. If this is also present, then the next step is the 3-carbon ceramide and the enzyme neuraminidase, because N-acetyl neuraminic acid is a sialic acid and is attached right here at the terminal group structure. So this is removed by the enzyme neuraminidase. And then it results into disaccharide, ceramide disaccharide, or glucose and galactose, or in other words, lactosyl ceramide. This terminal galactose can be removed by beta-galactosidase enzyme, and if this enzyme is deficient, then the lactosyl ceramide is accumulated, and the name of the disease is Krebs disease. And finally, if this enzyme is also present, then we have the glucose ceramide, which can be removed by beta-glucosidase enzyme. And if this enzyme is absent, then the glucosyl ceramide, which is cerebroside, is accumulated, and the disease is the Gaucher disease. These are the chart that shows you different type of lipid storage diseases. Coming to respiratory distress syndrome, this is the important part of the lecture. The term respiratory distress syndrome are most often applied to the severe lung disorder in neonates, which is primarily related to lung immaturity. It is responsible for more infant deaths and neurological complications. In these children, the breathing rate is usually more than 60 breaths per minute, and or use of accessory muscle of respiration, which may accompanied by grunting, a very loud type of disease of the sound of the lungs. This is because of the deficiency of this surfactant. Surfactant production starts around 20th week of the gestation of life and peaks up at 35th or 35 weeks of the gestation. Therefore, any neonate less than of 35 weeks is prone to develop respiratory distress syndrome. Without surfactant, infants are unable to keep their lungs inflated. Respiratory distress syndrome, the most common respiratory disorder in preterm neonates, once the major cause of mortality in premature neonates. The incidence and severity of respiratory distress syndrome is inversely related to the gestational age and birth weight of the infant. That is, if the gestational age is less than 35 weeks, then the baby is more prone to the respiratory distress syndrome. Acute lung disease of the newborn caused by surfactant deficiency. RDS is the clinical expression of surfactant deficiency and its histological counterpart, hairline membrane disease. Now, this tells you the mechanism, how it works. Phospholipids are produced by alveolar type 2 cells. Look right here, these are the right here, alveolar type 2 cells. Lower surface tension, if we have, then as alveoli, this is the alveolar type 2, and right here is the surfactant surrounded the type 2 alveolar cells. So what surfactant does? It reduces the surface tension right here of the alveolar cells, with the liquid of the alveolar cell. As alveolar cell radius, this one, decreases, surfactant's ability to lower surface tension increases, and vice versa. The half-life of surfactant is about 30 hours. Again, remember, as the alveoli radius, this one, decreases, then the surfactant lining, this one, increases the surface tension, and therefore lowers the surface tension, and the lungs can easily be inflated. The half-life of surfactant is 30 hours. How it works, you can write here. Collapsed alveoli and normal inflated alveoli. Phosphatidylcholine is the one which is the surfactant. Lung surfactant is 90% lipids, dipalmityl phosphatidylcholine, and plus 10% protein. So the surfactant is dipalmityl phosphatidylcholine along with 10% protein. Look right here. This is the inflated alveoli of the terminal sac. During inspiration and expiration. During inspiration, it is inflated. During expiration, it is de-inflated. Now, without the surfactant, without lung surfactant, sac collapses, as you have seen right here. Then, if it collapses, then the 10 times the normal pressure is needed to re-inflate it. So lung surfactant, what it does, it reduces the surface tension of water fluid lining the surface of the alveoli sac, as we have seen, in the previous slide, preventing collapse. So then, if the surfactant is present, then less pressure is needed to reinflate sac when surfactant is present. In the absence, then 10 times more pressure is required than normal for the reinflation of the alveoli. What is surfactant? And what is the function of surfactant? Function of surfactant: decrease the surface tension, to promote lung expansion during inspiration, to prevent alveolar collapse and loss of lung volume at the end of expiration. Surfactant is identifiable in fetal lungs as early as 16 weeks of gestation, though its proper secretion begins after 24 weeks of gestation and is synthesized most abundantly after 35 weeks of gestation. Pulmonary surfactants are phospholipids synthesized in the cell, in the type 2 cell lining of the alveoli surfactant. Cell lining the alveoli surfactant. Phospholipid produced by alveoli type 2 cells. They lower the surface tension. As alveoli radius, as I pointed out, decreases, surfactant's ability to lower surface tension increases. The half-life of surfactant is 30 hours. Thank you.